So welcome everybody, my name is Marta, and indeed I'm going to be presenting our project on developing a biomimetic glomerular filtration barrier for testing diabetic nephropathy. So we are going to be talking kidney. The kidney is the organ in the body that is in charge of filtering blood and removing waste products and excess of water. And we roughly filtrate like 180 liters of blood per day. And to do so, we have these small functional units, which are the nephrons. And basically, the nephron is like a long tube with different segments. And in each segment, there is a different cell type. And each cell type will take care of a different function, so whether it's reabsorption or refiltration, etc. The first segment of the nephron is the glomerulus. The glomerulus is basically a capillary knot surrounded by the nephron itself. And here is the main place for blood filtration. So everything that is in the blood will go to the inside of the glomerulus, and then through the rest of the nephron will get reabsorbed. To make sure that nothing that we want to keep in the body is filtrated, we have this. We have the glomerular filtration barrier, which is a very highly specialized, highly precise filtration barrier, which is composed by this one? Yes, which is composed by the podocytes, the glomerular basal membrane, which is secreted by podocyte and endothelial cells and is made of collagen type 4 and laminin, and endothelial cells. The podocytes, which are highly specialized epithelial cells wrapped around the capillaries, and then they form these very tight junctions that look like a zipper, and whatever needs to go out from the blood towards the nephron goes through this sleeve diaphragms. However, in disease, like diabetic nephropathy, there are a few processes that are not longer working correctly and lead to proteinuria. So for some of the things that happen is that you lose the glycocalyx components, the food processes, which are like these protrusions of the cytoplasm that create the zipper, are no longer functional, and they kind of fuse with each other. The glomerular basal membrane becomes thickened, and well, that leads, as I've just mentioned, to proteinuria and loss of other molecules. That was a very nice drawing, I hope. But this is how actually the glomerulus looks like in reality. So here we have the podocyte body and the food processes which wrap around the capillaries, and you can see here the secondary food processes and the slit diaphragms. In the literature, we've seen how other colleagues have proposed, or they try to increase the phenotypical signature of these cells in vitro, which is usually lost, as we know. And they use these half domes, and what they see is that the podocytes, the body stays like on the bottom, and the food processes come and try to wrap the balls, the half domes. So in this sense, we can somehow leverage the topography of our substrate to make sure that our cells are a bit more comfortable and perform a bit better. Besides the topographical features, some others have already tried to put flow on the system, so they have topographical guidance and flow. So our project is conceived within this mix of trying to enhance cell behavior with the topography, and we want to create a glomerular filtration barrier that has curvature. So what we are doing is we propose a 3D scaffold based on the hollow fiber membranes. These hollow fiber membranes come from the hemodialyzer that we use for dialysis. So in that sense, it's a material that we know is biocompatible and that it's already accepted and used in the clinic. So this hollow fiber is a tube, which is porous, and well, here it's very long. We make it smaller pieces, and it has a cutoff size of above 65 kilodalton, and is perfusible on the inside and the outside. And in my group, we have already proposed that this scaffold for gut modeling and for proximal tubule modeling. So it made a lot of sense that we kind of propose this for the glomerulus as well. So the idea is to combine this 3D scaffold with cells. And for cells, we're using a cell line of conditionally immortalized podocytes. And very briefly, I hope everybody knows how these cells work, but very briefly, cells have, they are infected with a temperature-sensitive virus. So basically, they have an immature stage in which they proliferate forever, if we let them. And upon the temperature switch, they enter cycle arrest and they stop proliferating and they express the mature markers. In this case, we go have nephrin and WT1 in this picture. So we have our fiber. First, we sterilize it, very simple, with ethanol for 1 hour. We wash away the ethanol. Then we coat it with L-DOPA, which is an amino acid that promotes cell adhesion. Then we further biofunctionalize the surface with collagen type 4 and laminin, which are components of the glomerular basal membrane. So it makes sense that for making the cell happy, we add these materials into the fiber. And of course, I will go a bit later into detail, but we also try a blend of the two components. Then we seed the cells. We let them proliferate for 7 days in this 33 degrees, so they are able to form this cell layer around the fiber. Then they mature for 14 days at 37 degrees, and after this period, we characterize our fiber. And we do that with a scanning electron microscope to see the cell disposition around the fiber. We do immunostaining to check, and qPCR for checking the specific markers. And because we want to have a filtration barrier and one that works, we also do barrier selectivity with fit-signaling for small molecule and different size dextrant, which will tell us where the sieving capacity is. I want to very briefly mention that for the immunostaining, we acquire the images, then we do a lot of cell segmentation with Cell Profiler, and we analyze the data with PRISM. And I will present results mostly on coverage of the fiber percentage, which gives us a value between the area stained and the total fiber area. So this is overall how our seeding protocol looks like. So now we're going to have a bit of results. So first, of course, things didn't work straight ahead. We saw a very interesting fact, and it was that the cells had a very high turnover ratio upon maturation. So what was happening is that when we had our cells before transferring to the maturation step at 33 degrees, they were covering very nicely the fiber. We had a cell layer which could have sieving properties because it's very covered. But when we transferred to 37, we saw that cells detach and very big gaps appear. So that's not ideal if we want to check for filtration. Because, here we can see how upon maturation, the number of cells really goes down, and the coverage of the fiber as well for the laminin and the collagen. So at the beginning, we checked both of these separately, but then we decided that maybe it made sense, or maybe we can enhance the coverage if we made a blend of the both of them. So as we can see in the 3 figures and the graphs, the blend of the collagen and the laminin improved slightly, or a bit more than slightly, the coverage on the fiber. So we were very happy to see that this combination of coatings worked. Then, okay, now we have a fiber that is fully covered. Good. We need to check whether the phenotypical markers of the mature cells were there. So for that, we check for cell arrangement with for actin filaments, for production of ECM components, laminin and collagen 4, and expression of podocyte-specific markers. So for the laminin, it was relatively simple to see that the cells were producing their own laminin because it was very properly located below the cells. However, for the collagen, that's a bit more complicated because the staining of the background is quite high. And we were also very happy to see that we have nephrin and podosine positive cells, and WT1, it's also positive, which is in other tissues is cancerogen marker, but in podocytes is a maturation marker. Well, I don't know if you can see that one, but we were very happy to see that it was positive. So likewise, we were able to check for the production of, or we were able to compare between the coatings on the production of these markers. And well, obviously collagen 4 we cannot properly quantify when it's alone. It's very difficult. And for the rest, we see that the expression of when there is a blend is still quite high. So right now we have a fiber that is covered by cells. These cells have their mature markers, but we still want to prove that we can have a filtration function. So for that, we have in our lab a microfluidic setup that is very simple, consists of a pump and a microfluidic chamber in which we place the fiber in the middle, and we can perfuse through the fiber. So the fluorescent moieties go from the inside to the fiber and then through the cell layer, and then we can sample the basolateral and the apical fraction to see how effective our filtration was. So this is still work in process, so the results are a bit mixed. This is how our setup actually looks like, and in here, very tiny, very small, you can see the fiber, and well, the yellow is all the things, all the fluorescent moieties that diffuse. And for that, until now, we have only tried with collagen-coated fibers, but we, of course, will extend to the other types. And what we see is that when we compare the leakage of the fiber, the collagen with our laminin control, like there is not a lot of difference between the coatings. However, they are able to, they let fluorescent moieties go out a bit more than when we have a fiber that is completely sealed. So that's good news. We also check for dextran leakage because the glomerular filtration barrier has a sieving size of 70 kilodalton, 77 kilodalton, so we would want the 40 kilodaltons to go through, 70 be a bit in between because we don't have one size, but a range of sizes in the moiety, and the 250 kilodalton do not go through. Here we add another control, which was a fiber with no cells, and we see that there seems to be a trend that the 250 don't go through, but of course we need to optimize. We need to repeat this for the optimized coating of the fiber. In parallel, we wanted to, because at the end we want to mimic diabetic nephropathy, so we went ahead and tried to establish the diabetic nephropathy conditions in 2D. So for that, we have our low glucose medium, which is the medium that we usually culture our cells. We had a osmotic control, and then we have several conditions with high glucose and some acids, and then we have a timeline in which we seed the cells, they proliferate until they form a monolayer in 2D, then we let them mature for 14 days, we starve them for 24 hours before injuring them for 48 or 72 hours, and then we do the readouts either in immune staining, or we also did a cell viability assay. So here, sadly, we saw that there is really not a lot of difference between any of the conditions. The cells, yes, they don't look very happy when we compare to those that were not actually starved. But there was, or either by the metabolic activity, there was not a very significant difference in any of the conditions. So we are a bit trying to still figure out what is happening and whether we can somehow promote this injury a bit further. However, we gave it a go in 3D because 3D cells are a bit happier, and what we saw is for the fibers is that most of the cells actually deattach. So this is something that we were not expecting because the injury didn't seem that bad in 2D, but here is really, really efficient. We need to further optimize the whole injury site, because at the end we want to have cells still there. Like, we know that the podocytes are not completely gone in diabetic injury, so we need to still figure out the conditions. But I want to finish the talk with some conclusive remarks. So first, we saw that upon surface biofunctionalizations, we avoided the cell detachment that was a bit tricky at the beginning, and we allowed the cells to establish this cell layer that will further allow us to do filtration experiments. We saw that the podocytes have a mature phenotypical signature, as shown by the mature stainings of nephrin, podocin, and WT1, and they are also able to deposit their own glomerular basal membrane, as we see by the stainings of laminin and collagen 4. We also see that up to a certain extent, there is already a barrier with sealing properties. We further need to optimize that, but until now, the results, we're very happy with how promising it looks. And we are still working on the diabetic nephropathy conditions, but mainly we see a very huge cell loss and the impaired glomerular muscle membrane deposition. We thought this. I think I'm a bit ahead of time, so that's nice. I would like to thank everybody who cooperated in this work, mainly Lise, who was a student with us and did a lot of the optimization of the cell culture, and well, the rest of my team, especially my supervisors, Ross and Sylvia, and the whole group of experimental pharmacology at Utrecht University. And now, if there are any questions, I'll be very happy to answer.
Thank you for your nice talk. So you want to mimic the curvature. So have you tried different diameters of fibers? Because maybe it's a bit too big for the podocytes to feel the curvature?
I fully agree with that, or that statement. Yeah, the fiber is quite large compared to the diameter of the capillary glomeruli, which are very, very tiny. This fiber, because it's commercial, we cannot tune it. So that's a that's a limitation. We are trying other approaches to be able to get smaller diameter tubes, but not based on this one. This one is a bit more useful for drug injury, toxicology studies, etc., because we have a higher throughput than whether we use any other chip or any other biofabrication strategies to create that tube. We are willing to forfeit that for a higher throughput here. But yeah.
Any other question?
First of all, thank you. Really interesting concept. Have you thought about adding glomerular endothelial cells to the model to improve the phenotype?
Yeah, that is also one of our next steps, to make a co-culture of the endothelial cells. So yeah, we will try first endothelial cells only in the fiber, and then a combination of both of them, and see whether we can indeed use the hollow fiber as a scaffold to further develop our, Thanks.
Okay, then thank you very much, Marta, for the great talk and the great work.